This talk is about the neurobiology of sleep. It is intended for the neurophysiology fellows at Nicholas Children's Hospital. It will be conducted in a question and answer format. The first question: the ascending pathway that keeps the forebrain awake originates in the rostral pons and caudal midbrain. A true, B false. Large part of the experiments that led to discovering the ascending activating system were conducted in cats. In this frame, I have enlarged the head of a cat to point to you the very important areas involved in the production of the wake-sleep cycle. The area is that of the upper pons and lower midbrain. So the answer to this question is true. Next question. The loss of sleep-wake cycles after a cut between the colliculi through the upper midbrain is permanent. A true, B false. A normal cat wake-sleep cycle is characterized by alternating periods between low voltage mixed activity waves and high voltage slow waves. A cut at the level of the upper midbrain converts the EEG into a monotonous rhythm characterized by high voltage slow waves. Yet, about 30 days after the experiment, the sleep cycling starts again. So the answer to this question is false. Next question. Arousal promoting neurons are present in the A pons, B pons and midbrain, C pons, midbrain and posterior lateral hypothalamus, D pons, midbrain, posterior lateral hypothalamus and basal forebrain. In this frame, I have listed 4 important regions involved in promoting arousal. As we can deduce from the answer to the first question in this talk, an important arousal promoting group of neurons are in the caudal midbrain and rostral pons. These neurons form 2 groups named according to their location as LDT and PPT. LDT stands for laterodorsal tegmental nucleus. PPT stands for pedunculopontine tegmental nucleus. The neurotransmitters they produce is acetylcholine. During this talk, I will often refer to this nuclei, that is to the LDT and PPT neuronal group, as the caudal midbrain cholinergic group. Axons from these neurons reach the thalamus, the hypothalamus, the basal forebrain, and the prefrontal cortex. A second bunch of arousal promoting neurons, which I have chosen to lump into a large group constituted by different structures, is located at the upper pons. The reason I lump them together, creating a multigroup, is because that is the way that is done in a significant number of articles. In this frame, I have listed the 6 groups that constitute the multigroup by their initial letters. They are all monoaminergic neurons. I will refer to them as the pontine monoaminergic neurons in the rest of this talk. The axons of these neurons have the same trajectory as those formed by the caudal midbrain cholinergic group. The third group of arousal promoting neurons are located in the posterior lateral hypothalamus. This group is constituted by neurons with different projections and neurotransmitters. One cluster of cells release orexin. Orexin is also called hypocretin. These orexin-releasing neurons connect with neurons in the basal forebrain, in the caudal midbrain, in the cholinergic group, and in the pontine monoaminergic group. The other cluster of neurons in the posterior lateral thalamus with arousal promoting qualities are glutaminergic. These neurons make contact with neurons in the basal forebrain. The fourth group of arousal promoting neurons is in the basal forebrain. They project to the prefrontal cortical region and use GABA and glutamate as their neurotransmitter. So the answer to this question is D. Next question. The cholinergic input to the thalamus from the laterodorsal tegmental nucleus and the pedunculopontine tegmental nucleus induce the thalamic reticular nucleus neurons to secrete GABA. A true, B false. The caudal midbrain cholinergic neurons connect with several nuclei in the thalamus. The thalamus, as you know, has several nuclei. Some of them can be seen from the outside of the thalamus. Others can only be seen when the thalamus is cut through the middle, as I have done in this figure. The arrow points to the reticular nucleus. The cholinergic neurons from the LDT and the PPT make a very important contact with the neurons in the reticular nucleus. The reticular nucleus of the thalamus neurons are GABA producers. So, when activated by acetylcholine, the amount of GABA released by the reticular nucleus increases. The result of this increase is that all the thalamic nuclei neurons become inhibited. Such inhibition blocks neurons that themselves block the passage of alerting signals to the brain, thus allowing sensory information to activate the cortex as a consequence of the secretion of GABA into the thalamus. Take a few seconds to look at this frame and try to understand it. So the answer to this question is true. Next question. Which of the following is not true regarding cholinergic neurons in the lateral-dorsal tegmental nucleus and the pendulopontine tegmental nucleus? A. Fire most rapidly during wakefulness and rapid eye movement sleep. B. Make connections with prefrontal cortex. C. Make connections with the cerebellum. D. Increase their firing rate just before the transition from a slow wave sleep to faster frequencies. Cholinergic neurons in the brainstem firing rate is highest during awake and REM sleep. They are the lowest during non-REM sleep. As previously stated, the LDT and the PTT neurons make contact with the prefrontal cortex, but they do not make contact with the cerebellum. If we plot neuronal firing rate versus time, we can see that going from slow wave sleep to low amplitude mixed frequency sleep, cholinergic neurons from the tegmental nuclei firing increases rapidly as the EEG changes from a slow wave sleep to low amplitude mixed frequency sleep. So the answer to this question is C. Next question. Which of the following is true regarding sleep-related monoaminergic neurons in the brainstem? A. Fire most rapidly during wakefulness. B. Decrease activity during non-REM sleep. C. Fall silent during REM sleep. D. All of the above. We already mentioned the pontine monoaminergic neurons and their connections. The pontine monoaminergic neurons, as we mentioned before, is a multi-group of neurons that are placed together for convenience during our discussion. This multi-group includes the noradrenergic locus coeruleus, the glutaminergic parabrachial nucleus, the glutaminergic precoeruleus nucleus, the serotoninergic dorsal raphe nucleus, the dopaminergic periaqueductal gray matter, and the histaminergic tuberomammillary nucleus. These neurons, meaning all the neurons in this multigroup, have the highest firing rate during wakefulness, fire some during non-REM sleep, and fire least or not at all during REM sleep. So the answer to this question is D. Next question. The posterior lateral hypothalamus arousal promoting neurons release all of the following neurotransmitters except A. Orexin or hypocretin, B. Serotonin, C. Glutamine, D. All of the above. The posterior lateral hypothalamus harbor arousal promoting neurons that release orexin, which, as you know, is also called hypocretin. These neurons, that is, these orexin releasing neurons, have their highest firing rate during wakefulness. The firing during sleep, whether it's REM or non-REM, is low to none. We also find in the posterior lateral hypothalamus intermingling with the orexin secreting neurons a group of arousal promoting neurons that secrete glutamine. These glutaminergic neurons have only contact with the basal forebrain. So the answer to this question is B. Next question. GABA releasing neurons in the basal forebrain produce cortical deactivation. A. True. B. False. In the cortex, excitatory interneurons acting on target neurons produce excitation. If an inhibitory neuron acts on an excitatory interneuron and inhibits it, the excitatory interneuron, which will then not release glutamine at the target neuron, will lead to the target neuron not being active. On the other hand, an inhibitory interneuron acting on a neuron will inhibit it. Now, if an inhibitory neuron acts upon an inhibitory interneuron, the interneuron would cease being active. Those will stop releasing GABA at the target neuron, and the target neuron will cease being inhibited. In order to see the effect of these actions, we must see what results from these events at the neural ensemble level, which I will explain in the following frames. GABA acting upon intermediary inhibitor interneurons will stop their inhibitory effect of the interneuron, thus decreasing the inhibitory load on the target cortical cell ensemble. This will result in an overall excitation of the target neuron ensemble, and in the case of the cortical neurons, that is, of the large cortical neuron cells, it will lead to EEG desynchronization and the production of low voltage mixed frequency activity. So the answer to this question is false. Next question. Sleep promoting neurons cluster are found in the A. Thalamus, B. Hypothalamus, C. Midbrain and pons, D. Pons. Sleep promoting neurons are primarily in the hypothalamus, which I have enlarged in this frame, as you can see. The hypothalamus consists of different zones that in an anterior to posterior direction have been labeled preoptic, anterior zone, tuberal zone, and posterior zone. A major sleep promoting zone is present in the preoptic zone. So the answer to this question is B. Next question. Which of the following neurotransmitters is released by the ventrolateral preoptic nucleus and the median preoptic nucleus? A. Galamine, B. GABA, C. Histamine, D. Acetylcholine. In the hypothalamus, in the preoptic area, there is a group of neurons called the ventrolateral preoptic nucleus, VLPO for short. These neurons release GABA and galamine at their target organs. The arousing promoter target organs are the caudal midbrain cholinergic cell group and all the pontine monoaminergic group. Neurons in the pontine neuroaminergic group and the caudal midbrain cholinergic groups send axons back to the VLPO. It seems that the VLPO has a mutually inhibitory relationship with the arousal promoting neurons of the caudal midbrain cholinergic neurons and the pontine monoaminergic neurons. This relation, that is the relation between the VLPO and the arousal promoting brainstem system that we have just mentioned, according to some, is the basis for the switching between sleep and awake states. Another important nucleus in the preoptic area that has the same action of promoting sleep is called the median preoptic nucleus. The neurons in this group also release GABA at their target organs, but not galanine. Neurons in this group will make connections with the VLPO. Notice the little arrow going into the VLPO that I have drawn in this frame. I am pointing it out to you by using the aqua arrow. The reason I emphasize this connection is because if and as the median preoptic nucleus secretes GABA, it would be reasonable to think that it would have an inhibitory effect on the VLPO, thus contrasting the arousal promoting effect of the VLPO. But this incongruency is present in many textbooks, and that's why I am expressing them here, so you be aware of it. Be it as it may, this nucleus, that is the median preoptic nucleus, also connects with rostral pontine monoaminergic neurons, except with those neurons belonging to the tuberomammillary group. A unique characteristic assigned to this mysterious nucleus is that it is only active after sleep has taken place for a few hours during what is called advanced sleep, and that according to some authors, this nucleus has a role of accumulating sleep pressure or hoarding the desire to sleep. So the answer to this question is B. So we have the arousal promoting system that we have previously described, and as you recall, it has 4 groups which are represented in this frame. If you have any question, just stop the video and look it over. We also have a sleep promoting system, which for our discussion is going to be constituted by the ventrolateral preoptic nucleus and the median preoptic nucleus. This frame represents a moment where arousal promoting neurons and sleep promoting neurons, that is the 2 systems, are in balance, which should take place for a very short amount of time, since the switching from one sleep state to the other is very quick. It is estimated to be done in a matter of seconds. Now I have added on your left a little face representing wakefulness, and below it another one representing non-REM sleep. To your right, I have represented neuronal firing rate. During wakefulness, the arousal promoter group are very active. During sleep, the preoptic region of the hypothalamus, which includes the ventrolateral preoptic nucleus and the median preoptic nucleus, will be very active, whereas those that were active before, the arousal promoter neurons, would be rather quiet. So the answer to this question is C. Next question. Which of the following neuronal groups discharge during REM? A. Sublaterodorsal nucleus. B. Precerulous region. C. Medial parabrachial nucleus. D. All of the above. The effector nuclei involved in initiating, terminating, and maintaining REM sleep are in the brainstem. They include the sublaterodorsal nucleus, also referred to as the subcerulous area, the precerulous region, the medial parabrachial nucleus, the lateral pontine tegmentum nucleus, and the ventrolateral periaqueductal gray matter. The laterodorsal nucleus, the precerulous region, and the median parabrachial nucleus send connections to the lateral pontine tegmental nucleus in the ventrolateral periaqueductal gray matter. These connections are both ways. These connections are also inhibitory in both directions. During REM sleep, the sublaterodorsal nucleus, the precerulous nucleus, and the medial parabrachial nucleus are very active. Thus, they are considered neurons with a REM on-firing pattern and are called REM-on neurons. So the answer to this question is D. Next question. Which of the following neuronal groups do not have excitatory input to the REM-off neuron? A. Orexin neurons. B. Noradrenergic neurons from the locus coeruleus. C. Serotonin neurons from the dorsal raphe nucleus. D. Cholinergic pedunculopontine tegmental neurons. The REM-off neurons are either excited or inhibited by other neuronal groups. The excitatory input to these neurons are from the orexin neurons, the noradrenergic locus coeruleus, and the serotonergic dorsal raphe nucleus. Remember, the activation of the REM-off neurons by all the excitatory neuronal groups we have just mentioned precludes REM sleep. So the answer to this question is D. Next question. Which of the following neuronal groups do not have inhibitory input to the REM-off neurons? A. Precerulous region. D. Melanin concentration hormone releasing neurons. D. Serotonergic dorsal raphe nucleus. D. Cholinergic pedunculopontine tegmental neurons. The neurons that inhibit the REM-off neurons are the REM-on neurons, which include, as we have previously mentioned, neurons from the sublateral nucleus of the hypothalamus, the precerulous region, and the medial parabrachial nucleus. In addition, other inhibitory elements are the ventrolateral preoptic nucleus, the posterior lateral hypothalamic melanin concentration hormone, and GABA releasing neurons, and the cholinergic tegmental neurons, which are the LDT and the PPT neurons. Remember that the activation of the REM-off neurons by all the inhibitory neurons group that I just had mentioned produces REM sleep. So the answer to this question is C. Next question. The only input to the REM-on neurons is from the REM-off neurons. A true, B false. REM-on neurons only have one input. This input is from the REM-off neurons, and it is, as we mentioned, inhibitory. So the answer to this question is A. Next question. The only REM-off neurons output is to the REM-on neurons. A true, B false. The only output of the REM-off neuron is to the REM-on neurons. So the answer to this question is true. Next question. The REM-on neurons output include all of the following except A. Cholinergic basal forebrain neurons, B. REM-off neurons, C. Medullary and spinal interneurons, D. orexin neurons. The REM-on neuron output is to the REM-off neurons, to the basal forebrain, and to the medullary and spinal interneurons. So the answer to this question is D. Next question. Dash neurons inhibit entry into REM sleep during wakefulness by exciting neurons in the REM-off population. A. VLPAG, D. orexin, C. VLPO, D. cholinergic. The sublateral dorsal nucleus, the pre-ceruleus, and the medial parabrachial nucleus are very active during REM. The neurons in the lateral pontine tegmentum nucleus and the ventrolateral periaqueductal gray matter are very active during non-REM sleep. Orexin neurons, also called hypocretin neurons, connect with the REM-off neurons. This connection is excitatory, so they activate the firing of the REM-off neurons. The activated REM-off neurons inhibit the REM-on neurons and thus prevent or stop REM sleep. So the answer to this question is B. Next question. Osserine neurons targets include A. pontine monoaminergic neurons, D. REM-off neurons, C. basal forebrain cholinergic neurons, D. all of the above. In this frame, I will limit the connections of the orexin neurons to those directly involved in stabilization of a sleep, wake cycle, and stabilization of REM. So as you can see, orexin neurons send axons to the basal forebrain, the caudal midbrain cholinergic group, the pontine monoaminergic neuron group, especially to the locus ceruleus, and the REM-off neurons. So the answer to this question is D. Next question. Dash neurons promote the entry into REM sleep from non-REM sleep by inhibiting neurons in the REM-off population. A. VLPAG, B. orexin, C. VLPO, D. glutaminergic. The neurons that promote REM sleep do so by inhibiting REM-off cells discharge. This is achieved by the activity of the REM-on cells. The ventrolateral preoptic nucleus, the melanin concentrating hormone and GABA releasing neurons of the posterior lateral hypothalamus, and the caudal midbrain cholinergic group? So the answer to this question is C. Next question. VLPO neurons fired at a rapid rate during non-REM and REM sleep. A true, B false. As you can see in this chart, the VLPO as well as the MNPO group of neurons have a rapid firing rate during non-REM sleep and during REM sleep. So the answer to this question is true. Next question. Orexin neurons excite REM off neurons and VLPO neurons inhibit REM off neurons. A true, B false. Orexin neurons excite REM off neurons, whereas ventrolateral preoptic nucleus neurons inhibit REM off neurons. So the answer to this question is true. Next question. Which of the following is true regarding lateral hypothalamic REM sleep active cells? A. They are mixed in with orexin neurons. B. They produce melanin concentrating hormone and GABA. C. They have the same targets as orexin neurons. D. They excite and inhibit according to the target organs. The posterior lateral hypothalamus has REM sleep preventing cells. They are the oxyrin neurons. These neurons, as previously stated, have a wide axonal distribution and are excitatory at their target organs. The same region has REM sleep activating neurons. The REM activating neurons in this region are the melanin concentrating hormone, which have the same targets as the orexin neurons but are inhibitory, and the GABA releasing neurons, which also have the same targets as the melanin concentrating hormone and are inhibitory. So the answer to this question is D. Next question. Which of the following is not true regarding melanin concentrating hormone producing and posterior thalamic GABA producing neurons? A. They are mixed with orexin neurons in the hypothalamus. B. Their functions are opposite to those of the orexin neurons. C. They innervate nearly all the same targets as orexin neurons. D. They promote arousal. In this frame, I have represented the orexin neurons and their connections. As we just discussed during the prior question, these neurons mixed with other type of neurons. The other type of neurons are the melanin concentrating hormone and the GABA releasing hormones. Melanin concentrating hormone and lateral hypothalamic GABA neurons fire rapidly during REM sleep. So their mechanism of action appears to be inhibition of the REM of neurons. This figure is a cartoonish representation of the firing activity of melanin concentrating hormones and GABA from the lateral posterior hypothalamus. This pattern of firing contrasts greatly with the pattern of orexin releasing neurons. So the answer to this question is D. Next question. Which of the following neurons discharge during REM and are the source of PGO waves? A. Locus coeruleus, B. PPT and LDT neurons, C. Dorsal raphe nucleus, D. Tuberomammillary nucleus. This frame features all the inhibitory neurons that contact the REM of neurons. These neuronal groups, by inhibiting the REM of neurons, produce REM sleep. Among these, we find the lateral dorsal tegmental nucleus and the pontopedunculary nucleus. The pontopeduncular nucleus appears to be involved in the generation of PGO waves. The PGO waves have been found in animals but not in humans. The PGO waves, as you can see in this frame, have been found in brains of cats, and as you can see, the arrows indicate their site of origin. So the answer to this question is B. Next question. Lesions in the LDT and PPT, in addition to suppressing PGO waves, suppress REM sleep atonia. A true, B false. This figure illustrates the effect of pedunculopontal lesion in subceruleus lesion. Remember that the term subceruleus and sublaterodorsal region of the hypothalamus indicate the same anatomical area. This study that I am showing you was done in cats and published in the journal Brain in 1992. EEG was recorded from the motor cortex. Eye movements were also recorded. Recording was also done from the lateral geniculate nucleus and also from postural muscles. This recording were done before the lesion, and such a recording was labeled normal REM sleep, as you can see in the top panel and in the lower panel. When the site injured was the pedunculopontine area, the result were the following: the EEG activity, which was consistent with REM before the lesion, kept consistent with REM after the lesion. Rapid eye movements no longer were present. PGO spikes no longer were detected in the lateral geniculate nucleus, but atonia remained undisturbed as it was before the lesion was done. So these findings support that the pontopeduncular area is responsible for the generation of PGO waves and extraocular movements. When the lesion was done at the subceruleus nucleus, the EEG activity was unchanged. Rapid eye movements remain unchanged. PGO wave remain unchanged. But REM sleep-induced atonia disappeared. So the subceruleus region is responsible for REM-induced atonia. The answer to this question is B. Next question. GABAergic neurons in the sublaterodorsal region are responsible for the production of atonia during REM sleep. A true, B false. The atonia that occurs during REM sleep is orchestrated by the neurons in the sublaterodorsal nucleus of the hypothalamus. The neurons that produce the atonia belong to a subset of neurons in the sublaterodorsal nucleus that release glutamine. They make connections with medullary interneurons and spinal interneurons, which interacts with lower motor neurons in the medulla and the spine, leading to their hyperpolarization. Muscles spared from the constant atonia during REM sleep are those from the diaphragm, the extraocular muscles, and the stapedius and tensor tympani muscles. So the answer to this question is false. Next question. Which neuron group triggers REM sleep? A. Oxygen neurons. B. Ventrolateral preoptic nucleus. C. Pontine monoaminergic multigroup. B. Basal forebrain cholinergic neurons. There are neurons that discharge during REM sleep. As you know, some of them are called REM-on neurons. I will, for the purpose of answering this question, limit them to the LDT, PT nuclei and the group we have been calling the REM-on neurons listed in this frame. There are also neurons that discharge during non-REM sleep. They are called REM-off neurons. Usually, most researchers restrict these neurons to those in the lateral pontine tegmentum and in the VLPAG, which stands for ventrolateral periaqueductal gray matter. it is easy to understand their relation if we do so as we did when we're thinking about the relation between wake and sleep state. That is, to think of them as a balancing act. During non-REM sleep, the balance will tilt as indicated in this frame. The REM-off neurons of the lateral pontine tegmentum and the ventrolateral periaqueductal gray fire at a very fast rate, whereas the REM-on neurons do not fire or fire very little at all. During REM-on neurons of the medial parabrachial nucleus, pre-ceruleus region, the sublaterodorsal nucleus of the hypothalamus, the lateral dorsal tegmental nucleus, and those in the pontopeduncular nucleus fire rapidly, whereas those from the REM-off neurons in the lateral pontine tegmentum and the ventrolateral periaqueductal gray fire at a very slow rate or not at all. But a proper answer for the question as to what triggers REM cells to start firing remains difficult to determine. We know that orexin neurons prevent REM sleep from happening during wakefulness. In the past, and still today by some researchers, the action of the preoptic nuclei neurons are considered the triggering event for REM sleep. Yet the fact that these neurons fire rapidly during non-REM remains a stumbling stone in understanding of this question as to what triggers REM. But those who believe that this is the triggering factor, they point out that these neurons also fire rapidly during REM sleep. Because of this uncertainty, that is that the VLPO and the MNPO neurons discharge rapidly during non-REM and REM, the eyes of most researchers has turned to melanin concentrating hormone and to the post-lateral hypothalamic release GABA neurons as the REM triggering neurotransmitters. Of the two just mentioned, melanin concentrating hormone appears to be a most likely candidate. Melanin concentrating hormone cells fire rapidly only during REM sleep. Their firing inhibits REM-off neurons. So the answer to this question is B. Next question. Which neuron group triggers REM sleep? A. Auxiliary neurons. B. Melanin concentrating hormone. C. Pontine monoaminergic multigroup. D. Basal forebrain cholinergic neurons. The only difference between the previous question and this one is that one choice in the answer was changed. This diagram, published in 2010, has a different explanation of what triggers REM sleep. But it points out that melanin concentrating hormone is the only one that actually triggers REM sleep. So the answer to this question, when it's made in this fashion, still will be B, melanin concentrating hormone. Next question. Which neuron group inhibits REM off neurons and wake on neurons? A, oscillin neurons. B, ventrolateral preoptic nucleus. C, melanin concentrating hormone. B, basal forebrain cholinergic neurons. Again, going back to the diagram published in 2010 in Neuron, you can see that in this figure they have melanin concentrating hormone as having a dual role. One is inhibiting the REM off neurons, thus triggering REM, and the other one inhibiting the Arousal Promoting monoaminergic group, which are wake on neurons that their inhibition would lead to sleep. But there is a problem with this diagram. The problem is that melanin concentrating hormone firing rate during non-REM sleep is very low. So their effect maintaining non-REM sleep appears inconsistent with their firing rate. Yet, having said this, a recent article from 2015 points out that our understanding of the role of melanin concentrating hormone neurons is incomplete, and that the role of this neurotransmitter in regard to sleep is increasing, at least our understanding of it. So the answer to this question is C. Next question. All neuronal group using the term ceruleus release norepinephrine at their targets. A true, B false. The locus ceruleus releases noradrenaline at its target. The subceruleus area releases GABA, and a subset of neurons in this group release glutamine. Preceruleus nucleus secrete glutamine. So the answer to this question is false. Next question. Which of the following is not true regarding adenosine? A not fatigued brain has low levels of adenosine. B, a fatigued brain has high levels of adenosine. C, adenosine activates most wake promoting neurons. D, extracellular levels of adenosine are governed by the hypothalamus. Adenosine levels rise with prolonged wakefulness, then fall during sleep. A rested or not fatigued brain has low levels of adenosine and high levels of ATP. I remember this fact by thinking about ATP as energy. A rested brain has lots of energy, so it has used all the adenosine to make ATP. A fatigued brain, on the other hand, has high levels of adenosine and low levels of ATP. A fatigued brain is using ATP and does not have time to make new ATP, and therefore the adenosine will be high. As you probably know, adenosine activates most wake-promoting neurons. But oddly enough, it is said that it disinhibits the ventrolateral preoptic nucleus, which, as you know, has a sleep-promoting function. Another interesting fact about adenosine is that its intracellular level depends almost completely on neuronal consumption, whereas its level in the extracellular space is regulated by astrocytes. So the answer to this question is D. Next question. Consolidated wakefulness increases the accumulation of homeostatic sleep drive during the day. A true, B false. The circadian alertness drive, or process C, if given free rein, fluctuates during a 25-hour period, a fact that is not represented in this frame. The suprachiasmatic nucleus is very important in the regulation of the circadian rhythm. Process C is influenced by time and travel. It is represented in this frame by the green arrows. There is a second process called process C. This process works in a very simple manner. The longer a person is awake, the more a person feels like going to sleep. The more the person is asleep, the less he or she wants to continue sleeping. This process, in addition to be called process C, is also labeled homeostatic drive. Process C is driven by external factors such as work and exercise. In this chart, it is represented by the magenta arrows. The balance between the circadian alertness drive and the homeostatic sleep drive determines how much sleep we get every day, or at least how sleepy we feel every day. And an important and interesting fact about it is that the sleep drive progressively increases with continuous period of alertness. So the answer to this question is true. Next question. The propensity to be awake is dictated by the circadian rhythm. A true, B false. Usually, in an average day, in an average man under relative normal condition, most propensity to be awake occurs in early afternoon and late in the evening. And the strongest propensity to be asleep is at 3 a.m. So the answer to this question is false. Next question. Which of the following parameters is not true? A. During REM sleep, brain oxygen consumption is higher than during non-REM sleep. B. During REM sleep, breathing responses to CO2 is decreased when compared to breathing responses to CO2 during non-REM sleep. C. During REM sleep, breathing responses to O2 is depressed when compared to the response of O2 during non-REM sleep. D. During REM sleep, core temperature is dependent on ambient temperature. Brain oxygen consumption is increased during REM sleep. Breathing response to CO2 is decreased during REM sleep. Breathing response to O2 is the same in REM sleep as it is in non-REM sleep. Temperature control during non-REM, the temperature control is homothermic. During REM, temperature control is poikilothermic. During REM sleep, mean temperature control is the same as snake. So the answer to this question is C. Next question. Loss of orexin neurons causes more frequent transitions into REM sleep. A. True. B. False. Orexin neurons bodies are in the lateral posterior hypothalamic region. Their axons have a wide distribution, as you can see in this figure. Orexin neurons stabilize the sleep-wake cycle in REM sleep. The loss of orexin neurons produces narcolepsy, which is characterized by a sleep-wake state instability. Falling asleep too often when awake, waking too often when asleep, and falling into partial REM states such as atonia when awake or dreaming when not quite asleep. So the answer to this question is true. Next question. Orexin neurons do all of the following except A. Fire most during wake and actively exploration. B. Stabilize normal waking state. C. Prevents transition to REM from wake state. D. Stabilize sleep stages. Orexin neurons do not fire during sleep. Hence, they cannot be directly responsible for maintaining sleep integrity, but they can be indirectly the cause of frequent night awakening when there is a deficiency of it. This figure summarizes the actions of the circadian alertness drive and homeostatic sleep drive during a 24-hour period. In the middle section of this figure, there is a section for time of the day. And on the right side, the ends of the arrow indicate the propensity to be awake or asleep. In a normal situation, when the wake period is consolidated to one period, the sleep drive increases progressively during the day, and then as we sleep, the sleep drive decreases. The homeostatic drive produces a sleep-awake cycle represented by the blue line in the middle of this figure. In the presence of orexin deficiency, as the result of intrusion of REM sleep during the day, there is no sustained accumulation of a sleep drive. Hence, night comes and there is multiple arousals as a consequence of decreased accumulation of a sleep drive. Therefore, the wake-sleep cycle in a person with orexin deficiency will have frequent arousals during sleep, as depicted here by the black line. This can be better appreciated if we compare with the wake sleep cycle in a normal person, represented by the interrupted blue line. So the answer to this question is D. Next question. Cortisol levels are highest early in the morning. A true, B false. This figure depicts the cycle of cortisol and melatonin secretion. As you can see, cortisol levels are highest in the morning and drop as the day goes by. They remain low during most of the night, but they start rising at about 3 to 4 a.m. and their peak is at around 8 in the morning. Melatonin levels are low during the day and rise quickly during the evening. Peak levels are from 10 p.m. to midnight. Growth hormone, contrary to cortisol, is highest in the first half of the night. So the answer to this question is true. Next question. A lesion in the anterior hypothalamus causes A. insomnia, B. sleepiness, C. both, D. neither. These three pictures of von Economo portrays the scientist, the soldier, and the visionary. This page is a photograph of the front piece of the renal publication where he described encephalitis lethargica. The area enclosed by the dotted line labeled A and shaded in green represents the sleep center as conceived by von Economo. The area shaded yellow is the area von Economo considered to be the sleep part of the center, and the area shaded aqua is the area von Economo considered to be the waking part of the center. A lesion in the sleep part produces insomnia, which is the anterior part, and a lesion in the waking part produces hypersomnia. So the answer to this question is A. Next question. Which of the following you would expect in a patient with narcolepsy? A. HLA DQB1 positive, B. low orexin CSF level, C. MSLT with 3 short onset REM sleep periods, D. all of the above. You can see in this frame the representation of the hypothalamus corresponding to a normal patient and a patient with narcolepsy. The density of the dots represent the number of orexin neurons. There are many in the hypothalamus of a normal individual and few in the hypothalamus of individual with narcolepsy. This can be appreciated in the photomicrograph below, color matched with the diagram above. The microphotograph of the normal person has many darker dots representing orexin neurons. The microphotograph of the narcoleptic patient has very few. Patients with narcolepsy are often HLA TQB1 positive and during MSLT have frequent short onset REM sleep periods. So the answer to this question is D. Thank you.